nutrition. So what I'm trying to answer today, at least partially, are the following questions. What is uncultured microbiome data? How do you characterize microbiome composition statistically? And which statistical methods were used until now to associate nutrition at the nutrients or food group levels to gut microbiome composition at Taxa level? Now, the microbiome is very complex thing, so we can see here that it has different location. We can find it in stool, cheek, tongue, nose, vagina, skin. Each location has different abundances of bacteria, and for example, here in the gut, bacteroids are the most abundant, and E. coli are also presented in the gut, but they are actually present very low abundance. So how do we characterize this? How do we recognize this abundance with the help of sequencing techniques? We are doing this with the help of metagenomics. This is a relatively new, it's a new science which is fast developing now due to the fast developing sequencing technologies. And a metagenome is a whole genetic material, DNA or RNA sequences, which are recovered directly for environmental samples. Now, metagenomic assesses genomic composition and diversity within and across microbial communities by means of sequencing technologies. And as I said, you can have targeted RNA gene sequencing, like for example for bacteria 16S, or whole metagenome shotgun sequencing. Now I will present today the 16S rRNA gene-based studies. So this means that we are getting, in this method, we are getting 454 short sequence reads of some V1, V9 regions of a gene in our sample. Now, this gene is used as a kind of a fingerprint for the taxa, meaning this is due to its ubiquity, to the fact that it has conserved regions and also very variable regions which allow, as I said, to identify the exact taxon. So, for example, here we can see kind of gene sequencing for the region V1, V2, V3. So these are the regions with high variability. The one between, they have low variability. So this allows us to identify how these sequences are stopped. And what we get from this technology is a lot, millions of nucleotide sequences, and we want, with the help of these sequences, to recognize the taxa abundances in our sample. Now, how do we do this? Well, I remember the taxonomy is a classification of organisms. At the beginning, Klaus Linnaeus had actually introduced this taxonomy as a concept for species, and it was we have clearly species, genus, family, order, class, phylum, kingdom, domain. They are very well defined for the Animalia domain, but for prokaryotes, there is no official classification and no firm estimate on the number of prokaryote species. So actually, there are over 7,000 species of bacteria and archaea known, and they are still discovering new species due to new technologies. meaning that we need to actually the scientists needed to redefine what's a taxonomy in this case, because they cannot rely anymore on what was called physical properties of the organism. So we actually think of taxonomy as the classification of microorganisms based on genes similarities or differences. And this means that we have these DNA fragments, as we saw in the 454, from the 454 sequencing technology. We have incorporated them in a library, and then a small proportion from the library is sequenced, and the reads are then binned into features called OTUs. So these are clusters of sequences which have different similarities, and based on these similarities we actually define what's family, genus, or species. This means that family has 85% similarities, genus 90%, and species 97% similarity for bacteria. After we did this, what happens? So we have again some thousands of OTUs in one sample, and then these are mapped to taxonomic profiling, and then all these appearances of each OTUs in one of these taxonomic classes is added, and you get what's called an OTU table. This means that here we have 19 OTUs in, for example, Cropococcus, or in bacteroids 23 in the first sample. So we get a table where we have the columns as taxonomy classes, and each line it actually corresponds to the structure for one sample. In our case, was this table is taken from a PopGen cohort, meaning that it's a human gut microbiome. Now, what I would like to characterize is to link, to associate, it's this taxa counts at, in this case, genus level to the food intake in PopGen. And also, when it's possible, also to some other socioeconomic characteristic like age or social status, or some health information about our individuals, like smoking status, health status, and so on. So how do you do this? So normally you would think about the regression method, and you are right. So here you have some presentation of the nutritional and the microbiome data. So on the left part you can see the Pearson correlation coefficients for here for the nutrients, and here for the food groups which we used. And you can see that the nutrients are highly correlated, at least some of them, and in the case of the food groups we have much low correlation. In the microbiome data, so what we understand that as a microbiome data is actually this OTU table. This is what's called taxa abundances. You can see that you have a lot of red and some white points here. This means that you have a lot of zeros. And so this is one first difficulty when we work with microbiome data. You have what's called statistical sparsity. Now, you have here correlated data, you have here sparsity. What can you do about it? Well, in a paper from Chen Li, there was given a solution which allows it to link each taxon to each food item, and this is done with the help of what's called sparse Dirichlet multinomial regression, and this is used for a variable selection. what do they do in this approach? Well, what are the steps which you should follow in the case if you work with data? Well, first would be to choose the probability distribution for the count data, then choose the link functions that connect the count to the covariates, and in the end, choose the penalty function to get the sparse estimation. So the third part might be newer to you, but the first two, I think everybody who worked with data knows that you need to think about it if you think about generalized linear models, for example. So now we try to answer the first question. One method used until now to deal with these counts is actually to transform them and to transform in proportional abundances. Another possibility is to rarefy them, and this means to throw away sequences so that all have the same size, because you have high heterogeneity between the counts. But both these so-called normalization methods lead to omission of available valid data, and this is a no-go in statistics. So that's why lately a lot of statisticians thought about how to use mixture models for the count data. So if you have counts, normally you think like something like Poisson multinomial distribution, but if you try to fit a Poisson or a multinomial model to the count data, then you get high over-dispersion. So that's why you actually would need to put some more variability on your Taxa proportion, and that's why you use what's called gamma Poisson or negative binomial or multinomial Dirichlet. So to exemplify what's this over-dispersion, I can show you here. You have real data on the right part, and then if you try to fit only multinomial, you can see that you get something like this. And it's not nearly, I mean, even visually you can see that's not nearly next to the real data. And that's why you try to add more variability in this part of the data. So this means that you go for the Dirichlet multinomial. What's this distribution where it's adds an additional variance term governing the over-dispersion to the multinomial variance. And then you get something like this. Maybe it's not perfect, but it's, I mean, you can also test it statistically with the LRT likelihood ratio test, but also visually you can see here that you get better results. So that's why it's recommended to use a kind of a mixture model. Now, mathematically it makes sense, and it makes the computation easier if you add what's called the link function. So instead of considering a beta coefficient which links each taxon with each covariate, you take actually the logarithmic link function, and you have actually an exponential relation between these two, between covariates and taxon. And luckily it makes sense also from biological point of view, because microorganisms develop exponentially in a positive, in a favorable environment. Now, you get to the M regression. So you see here you want to estimate this beta coefficient like in any regression, which represents actually your relation between the covariate and the taxon. The next step might be to try to do multiple testing or to use a likelihood ratio test. But in the first case, you have you will test only the association of a covariate with the microbiome composition, and in the second, you have the problem of the sometimes, I mean, a lot of times that the number of covariates you have is much higher than the number of samples. For in our case, for example, we had here we had something like 19 Taxa counts and plus and 42 food groups. This means that you have over 800 betas, 800 coefficients, and you have only 757 samples. So this is not very feasible. And that's why the statisticians are trying to use variable selection methods. And one very well known is lasso. This minimizes the minus log likelihood subject to the sum of the absolute value of the coefficient being less than a constant. Why did I put this? Well, because this constant actually makes you to put some coefficients to 0. So this condition makes you to put the coefficients to 0, and the existence of this constant actually it's given by what's called the tuning parameter. So it's a tuning parameter, and this is something which you need to take in consideration to calibrate if you use penalization methods. Now, we would like to have, we try to put something like this kind of a sparsity condition on all betas, or we can look at them at a group, meaning that we would like, for example, to have a sparsity in the group of nutrients, or in the group of food items, in general of the covariance. And that's why there is not only it's a lasso, there is also what's called group penalized lasso as a method. And also, if you would like to have a double sparsity, meaning that you have a sparsity in the group and inside within the group, then you have what's called sparse group lasso method. So this puts two conditions, and this allows you to get the sparsest solution at the group level and within group level. This makes sense because you would consider all the coefficients for one group, food group, or one nutrient as a group, and then we assume that many food items or nutrients have no effect or ignorable effect on the microbiome. Now, how do you calibrate this method? I mean, as I said, you get this tuning parameter, and you would like to calibrate. Well, a very well-known method is the Bayesian information criterion, BIC, and this describes the loss of information compared to an exact model. Usually its value has no meaning in itself, but it needs to be the lower, and it has some disadvantages. So it relies on a proper estimation of the degrees of freedom, is usually derived for large samples, and of course assumes that the model is correct. Another calibration method is cross-validation. So if you want to do a prediction problem, then since this cross-validation actually estimates the expected prediction error, it's a preferred criterion in prediction problems. But it also has a disadvantage that the variability of the selected model due to the random assignment of the observation to the false makes it a bit unreliable. So another method is stability selection. This uses as a principle for the selection of the tuning parameters the rates of the false discoveries, and it's actually a subsampling method. Now, what's specific to penalization methods until now is that they don't have p-values and things like that. So there is not inference theory in the sense of how you got used to it for statistical, I mean for parametric statistics, and this makes a bit difficult to get used to it. So usually to validate it, of course, if you want to validate your own method, like statisticians usually do, you can simulate your data and then compute the recall, the precision, or F1. But this works only for simulated data. For the reality, you need to, there are two methods until now. So first is to refit the final Dirichlet multinomial model, and then considering as a normal regression and compute the p-values and the coefficients of determination for this. But of course, this has a disadvantage that this method actually overlooks everything what happened until now. So this means that, for example, the computation of degrees of freedom is unclear, and it might overestimate first type error. And another method which is statistically grounded is bootstrap the data and compute the bootstrap selection probability. And if you get reliable selection probability, then you consider that you get a significant result. I mean, you have a significant coefficient. Now, just for fun, I show you how this would look like, so how this method would work with simulated data. So I considered taxa counts simulated for a Dirichlet multinomial, and then the food intake, and also some what's called categorical variables, gender, smoking, income status, for example. You can think about it. I took 50 covariates, and between these three categorical regressors, I took 20 taxas, and I consider that you have 4 significant covariants and 4 significant taxa. Then the food intake also simulated as a normal distribution, and I used a sparse group lasso with a big for variable selection. And so this is how you would actually report your results if you are in if you use a penalization method. So practically you have here only the what were non-negative, non-zero coefficients. These are the taxa here, and here the food groups which were detected, and the blue ones are the ones who actually are correctly identified, and we see also we have here false negatives and a lot of false positives. So this is something which you need always to take in consideration, that big inflates the number of false positives. Now, of course, this is just for fun, and but I will recommend you, if you work with data, to try, after you decide to use some distribution and some methods, to try to simulate data which look like your real data, and then to look how your method works, because this might tell you a lot about, for example, in this case, as I said, about inflating the number of false positives. Now, what I wanted to present today was how you can work with penalization methods in gut microbiome food or nutritional data analysis. These penalization methods are dealing with the case of a larger number of covariates, larger than the sample size. They are also lasso, and the group of methods around lasso actually are doing variable selection, and this means that they are also appropriate for the case of sparse data. Now, there are some heuristical but also some statistical funded approaches that allow to use this for the validation of the method. And in case of over-dispersion, you should look at the mixture distribution. And there is research underway for the inference theory for lasso and penalization methods, and I hope that in the near future we get other validation methods also. So, thank you.